probably answered the wrong thing but got it right, it'll rescore and say, ha, it's now wrong. So there was somehow a couple of the questions options didn't appear and it threw the order out, but only 5 people took it last night. So I can see how many people have taken the quizzes. I don't see who took what, but I can also see, which is really useful, what everyone answered. So I can really easily look at every question and see what people are having trouble with. So definitely take those if you have the time, even if you just take a minute or two to do it, because it'll really help me what are the topics that we need to spend a little more time on and what have you really got down. So some of them, there's 4 answers and 25% of you choose each given option. Well, okay, we have to spend some more time, and some of it, like 100%, get everything correct. So that's just a little plug for the quizzes. All right, so the good news is that everything we're going to talk about today can really be summarized in like 6 slides. So it's really not that much material. The bad news is I'm going to take 60 slides to talk about these 6 slides. But keep in mind that there are 6 simple slides there that will help you get through it. 2 of them are just on the case study, so just summarizing what the really key points are. And then everything else is just sort of going into a little bit more detail about that and giving you an opportunity to ask questions and whatever else is important. I'm just going to turn that light on. So our case study is a diabetic man. He has type 1 diabetes. Who can tell me the difference between type 1 and type 2 diabetes? Jeffrey.
Type 1 diabetes, you don't make insulin, whereas type 2 diabetes, your cells are resistant to insulin.
Good, that's a really great summary. So 1, you're not making insulin. 2, you're not sensitive to insulin. 1 is typically an autoimmune disease where you kill the cells that produce insulin, and there's a fairly easy treatment, which is delivering insulin to the individual. Type 1 used to be known as juvenile diabetes because typically its onset would be earlier in age, whereas type 2 diabetes would hit you later. Type 2 diabetes has an incidence up to 20%, which is pretty significant if you consider the complications are things like amputation, blindness, and death. It has a 100% cure rate by diet modification, but that's very difficult to actually enact in practice. So it's sort of an interesting condition that you will see a lot of. And this case study refers to one of the side effects, which is development of ulcerations that will often require amputation of a limb. So we have an older gentleman who has type 1 diabetes. He's had it for a long time. Presumably he's been trying to monitor it and take insulin, but over time it's not really going to be perfect. Maybe he's not really good at monitoring his blood glucose and taking insulin. So more recently he started to have tingling and numbness in his feet, and to a lesser extent his hands. And you're actually able to find a small ulceration in his foot. So no loss of motor function, and the deep tendon reflex is normal. So we'll talk about reflexes in a few weeks, but the reflex for now is normal. So at the end of today, what I want to do is review everything we've covered and consider why we have tingling, why we might have ulceration, and why some of these other aspects are still intact. So that should all be clear at the end of today. Today, we're going to talk about how action potentials are really initiated. So we've talked about action potentials, and we give a 5 millivolt depolarization in the lab, and that reaches threshold, and then you hit an action potential. I'm sure you're all shocked. You're shocked that all of your action potentials aren't initiated by 5 millivolt depolarizations initiated in the lab. So they have to come from somewhere, and they come from somewhere called receptor potentials. So today we're going to talk about how action potentials are generated from your sensory system. So our learning goals today, we're going to talk about how different signals from the environment, sort of getting an echo here, so I'm just going to close the door. It's bad enough to have to listen to myself once, 2 or 3 times is too much. So how is a sensory signal translated? Oh my gosh, look at that. Thank you. That's why he's the tech guy, right? He can notice things like that I miss. So we're going to talk all about receptor potentials. So today is mostly about receptor potentials. We're going to talk about a few different types of receptors. So basically our somatic receptors, so our skin receptors and some of our organ receptors, some receptors for temperature and distortion, and also for pain. The soccer team was named for the most potent pain-inducing molecule ever last year. So or maybe it was 2 years ago, substance P, which we'll talk about later. So that was the awesome soccer team name. Fast and slow adaptation, so we'll talk about what that means. And then how we code for different features. Intensity, so how do you detect the intensity, how bright light is, or how loud a sound is, or how just how intense something is. The location, so you can detect the location of something. Rats can smell in stereo, so they can smell where something is because their nostrils are wired that way. Quality or modality, so you're seeing something, but let's not try this. If we were to poke your cornea with a pencil, you would see light, but you're not really seeing light. You're just artificially triggering your cells. Don't try this. And temporal quality, so how long does something last? So the initiation and end of something. And then we're going to talk about pain and types of pain and fiber types. So that's what we're going to go over today. And we have to kind of zigzag around a little bit. So just bear with me. We have to present some details and then go back and forward. So our big problem is, how do we take these stimuli from the environment and turn them into some sort of useful information? So think of all the different things in the environment that you have to contend with. So mostly we're visual, we're auditory, that's our species' primary senses, so touch and smell and olfaction. We'll talk about those also, but these are really different. So photons coming out are really different from sound waves. So how are we taking those different features from the environment and turning them into some sort of information that our brain can process? And it turns out we use the same mechanism for everything. And that mechanism, what is it? Can anyone? We're going to have some action potentials. They're going to be there. We're going to have some receptor potentials. We're going to have some generator potentials. So you're going to change membrane conductance. So you're going to have a change in membrane conductance, and that change in membrane conductance to ions underlies everything. Now, which ion the conductance is changing to can vary, so there's some exceptions. And first you would start with what's detectable. So we detect light, chemicals, movement, touch, pressure. This is a platypus, so it's sort of a funny egg-laying mammal from Australia. They can detect electric fields, so they have electric field detectors in their beak, and they kind of wallow around in the mud and detect little critters. So sharks can do that also. Birds can detect magnetic fields, so you wouldn't need GPS. You could just detect a magnetic field and go south or something like that. Maybe that would be so good. We still need, I think, GPS. So anything that's really useful to detect, we can detect. Now, magnetic fields and electrical fields aren't so useful for us, so we don't have structures to detect them. And basically, we have 3 stages here. And this is the first time I'm going to show action potentials like this. So I just want you to be really clear how to read these. So the first thing we have is a stimulus. So we have our generic stimulus. It's turning on, and it turns off. And we're just measuring the intensity of the stimulus here. Now this stimulus is going to cause something called a receptor potential. A receptor potential, as you'll see written out on the next slide, is a local graded change in conductance. So local graded change in ion conductance, and that's represented here. So this would be deflection from resting membrane potential. We could imagine our resting membrane potential is minus 70 in our receptor structure, and the stimulus is causing some sort of receptor potential. They will often have these complex shapes where they are a little stronger and then they plateau off. And this is going to give rise to a series of action potentials. You'll see I will often represent action potentials as just a line. And that series of action potentials I'll call a train of action potentials. So this would be a series of action potentials. We have a stimulus. That causes a receptor potential, and a receptor potential is what? Local? Graded? Yeah, and it's a change in membrane conductance. And that's going to give rise to a series of action potentials. Maybe it'll give rise to no action potentials, just depending on what it is. So this is our generic case. And I love the little exceptions, and so I'll focus on those, but I'll make sure to point them out. So like I said, these receptors transform some stimulus into a receptor potential, and that's local and graded. So you have stretch, you're stretching, and that stretching is actually going to pull open ion channels. So you actually have ion channels you can just pull open, and that's going to give rise to receptor potential, so a change in ion conductance because you're pulling those channels open, and that, if you reach threshold, can start to fire some action potentials. So the receptor potentials are the result from a change, or they result in a change in membrane conductance, and the recovery isn't mediated like an action potential. So you just sort of diffuse out the change. So the receptor potential is generally pretty small. And it's not like the action potential. So what happens with the action potential? How do we recover from an action potential? Potassium, yeah, and where's potassium moving? Out of the cell, yeah. So you have some potassium movement, and that's going to repolarize you. So the structure of the receptor itself is pretty important for this process. We're going to talk about a number of different receptors. So I just want to focus on sort of a generic receptor. So in a generic receptor, you would have a receptor region. So this is the region with receptors that detect some stimulus. You would have something called a spike generating region. So a spike in this case is an action potential. And then you would have a conducting region, and that conducting region would transmit an action potential down, say your axon. So the receptor area itself doesn't have voltage-gated channels. So it doesn't have those voltage-gated sodium channels. Why would you not want voltage-gated channels here? Need louder. Nicholas.
Every stimulus is electrically based, I guess. I don't know.
What if you had some voltage gated channels there? What if you could kick in an action potential there? So if you don't have voltage gated channels, you can't kick in an action potential. So what if you had those here?
You wouldn't be able to see it.
So I hear a lot of the right answer, so that's good. So if you had voltage-gated channels and you had action potentials there, you couldn't have graded. So you couldn't have graded stimuli, like everything would be just as intense because your action potential, remember, is all or none. You have an action potential and it happens or it doesn't happen, and if it happens, it's this big, big depolarization and repolarization. The receptor potentials are not. Remember, they're local and they're graded. And because they're local and graded, we can detect a lot of different things about the stimulus. So this would also prevent the stimuli from summation. So for example, if you have two stimuli, they can add up. Their change in receptor potential can add up, and that can cause the cell to pass threshold. So by not having voltage-gated channels there, you have a lot of additional features from the stimulus that can be encoded. So really, today, until we get to pain, the focus is how these different stimuli are encoded into action potentials and how you get information out of that. So you have a receptor potential. I just told you there's no voltage-gated channels there. So how do you actually get an action potential? So that's something to consider. And this just goes back to our friend, the movement of current. So remember, anytime you have a change in current, it's a closed system, so you're going to draw off charge from adjacent regions. So if you have a receptor potential, say at the end of this sensory structure, it's going to be local and graded. You'll get some depolarization. You'll pull charge off this adjacent region, and then that can trigger your voltage-gated channel. So you have a receptor potential. It's a small change in the membrane resting potential, and that will basically travel down the structure in the same way the axon will conduct the action potentials down until it reaches more voltage-gated channels. So you have a small depolarization. It's going to draw charge off the adjacent region, and that's going to trigger the voltage-gated channels to open. This would be an example would be maybe sensory receptors in your skin. So you have sensory receptors in your skin. They have little detecting fingers out here. The cell body would be about here, and then you send the information on to the spinal cord. So you don't have voltage-gated channels there, but they're going to be close. So they're going to be in proximity, and depending on how large this receptor potential is, that influences how much charge you're going to draw from here and how long you're going to be drawing charge from this region. And that, in turn, will give you a different number of action potentials. So we'll see that that's how intensity can be encoded. So if you have a more intense receptor potential right here, it will continue to draw charge off for a longer period of time and send more action potentials, or more intense. The train of action potentials will be more dense, depending on the system. And so this is just an explanation of that. You're drawing charge off the adjacent region, and if it's enough to cause these voltage-gated channels to open, then you'll induce the Hodgkin-Huxley cycle, and that receptor potential will be transmitted on. So that's the basic of the receptor potential. I've mentioned that they're all due to changes in membrane conductance, so this is really key, but what that exactly is can be different in different systems. So we'll see that in the auditory and visual systems, these are very different than in the rest of the nervous system, and we'll talk about those in detail starting next week when we talk about the auditory system, and then the week after with the visual system. So today we're really going to focus on the somatic, the body receptors. And so we can consider these different receptors based on their location. So one is just, where are these? So we have a type of receptor called exteroreceptors. So they're on the exterior of the body. So they're going to arise from stimulus that come out from outside of the body. And these are some of the stimuli that they would be sensitive to. They include the special sense organs, so our eyes and our auditory system and our olfactory system. But those will each get their own treatment. So if we have exteroreceptors, we better have interoreceptors. So we have receptors that are located within the body. These might be in the viscera. What are your viscera? Your organs, and your blood vessels. So you need receptors in your blood vessels to see how far they're stretched out, and then those can give feedback to regulate blood pressure, for example. So these might be sensitive to chemical changes, to stretch, to temperature changes. So exteroreceptors, interoreceptors. Proprioceptors are a special class of interoreceptors. So these are interoreceptors that tell the brain where your body is in space. So these would be your muscle spindle organs, that'll be a slide or two from now, your Golgi tendon organs, so they're located in your joints and their muscles, and they feed eventually into your cerebellum and help your body regulate movement. So those are your proprioceptors, so they're constantly telling your brain where everything is, even if you're at rest. So your receptors can also be classified depending on what the stimulus is. So you can have mechanoreceptors, thermoreceptors, photoreceptors, chemoreceptors, so those are all pretty basic. You can pretty much just read the word and figure out what you're detecting. And then nociceptors. So nociceptors detect pain, and I always think like, ouch, no, that's a nociceptor. So you don't want that. I don't know how you'd remember it in other languages. It'd be hopeless. So again, this is just basically how we would classify some receptors. And also keep in mind that some cells contain a receptor structure and will transmit that signal on. Whereas some receptors are actually specialized structures or specialized cells. So you might have a cell that acts as a receptor structure and it sends the information on. And this is really important if you're working in regenerative medicine, because if you damage or destroyed a receptor cell, and you can replace the function of that receptor cell, like with a cochlear implant, well, that's a pretty easy solution. But if your receptor cell is also the cell that's transmitting information to the brain, and you destroy that whole structure, well, that's harder to deal with because you can't just apply stimulus. You have to somehow get that to the back of the brain. And we'll see in detail when we talk about the special senses, which those are. Here's a generic cartoon. This is our generic cartoon of a cell. Again, we'll have lots of different variations on this. So you have receptors that detect stimuli. That gives rise to a change in conductance, so that's our receptor potential. Sometimes you'll see generator potential in place of receptor potential, and the difference is a generator potential can generate action potentials, but doesn't always, and the receptor potential is a change, but we've settled on receptor potential, so that's what you'll see. So the receptor potential is going to be this change in conductance. It's a small, locally graded change in your resting membrane potential, as you'll see. And in our generic case, a generic case might be the auditory system. Somehow this is going to cause a change in calcium at the terminal, and then you're going to release neurotransmitter. So this is our generic cell. And you're going to see many variations on it as we go through. Like I mentioned, the sensory receptors themselves can have really different complexity. So you can have free nerve endings in your skin. And free nerve endings like to detect things like pain. Like if they get cut, they're going to start to fire. If they get damaged, they're going to start to fire. You can have organized receptors, so capsules and other structures that are good at detecting vibration or pressure. We'll talk about a few of those in a moment. And then specialized receptor cells. Those would mostly be the special senses. And we'll cover those in their own unit. And today we're going to focus on these two. Everyone with me? Everyone okay? Do we need a stretch break? Yeah? No? I'll have to put in some exercise. You know, which receptors am I activating? Where are they located? So we're just going to parade through some different types of receptors, and eventually we're going to come back to how these can mediate things like intensity. So one example would be receptors for temperature detection. And you're going to see this kind of channel a lot, TRP channels, which stands for transient receptor potential channels. And these channels, they will fire, they will conduct ions differently depending on their temperature. So some of them will open if they're heated. Some of them will be more open if they're cold. So this is the basic way that we detect temperature. Some of them detect multiple different things. We have a channel called TRP-M8. So your TRP channel, M, we'll see, means metabotropic, and 8 is just number 8. And it's sensitive to cold and menthol. So this is why menthol cigarettes seem nice and frosty. They're really not. You're just triggering your TRP-M8 channels. Or why if you take menthol cough drops, it seems cool and soothing. You're just stimulating these channels that are normally triggered by cold. This is just a rough graph of two different channels, so they have different temperature selectivities. So if you have cold channels, they're going to be active maximally around 25 degrees Celsius, which is pretty cold, like room temperature. They'll be shut around body temperature. And because as you cool down, molecular movement slows down, they're going to fall off as the temperature gets more cold. The heat-sensitive channels are going to peak around 42 degrees. Actually, at this temperature, you'll start to activate these channels again because you're starting to damage them. So sometimes heat can feel cold, and that's because you're damaging some of your cold receptors. So those are receptors for temperature. This would be how a pit viper could detect heat in its prey. It has a little organ, and it can just see heat. If you put your hand up against the stove, this is how you're detecting heat as one example. So the TRP channels, again, you're going to see them all over the place. Another type of TRP channel are the mechanical distortion channels. These are really simple. You just pull them open. You pull them open and sodium moves through, or in some cases potassium moves through. So your receptors for mechanical distortion are TRP channels, so think of stretching. When you're stretching, you could be pulling these channels open, or you could be squishing them shut. So really, really simple here. So that was temperature and mechanical. And what I'm going to switch now to is the receptors in our skin and the receptors in our muscle and our joints. Is everyone fine up to now? Does this pretty much review how much, some of you have probably seen a lot of this before and maybe some not so much. I guess especially the next section. You'll see it in histology. Maybe you've done this. What are you doing in histology right now? Connective tissue. All right, maybe. So some types of receptor structures, Merkel's discs, Meissner's corpuscles, Pacinian corpuscles, interoreceptors, we'll talk about muscle spindle and Golgi tendon organs, and then free nerve endings that mediate pain. That's what we'll finish with. So I'm going to cover these a little bit, and then those, and then we'll finish there, just to give you a quick outline of what we're doing. These are just an image of what these would look like histologically. So if you look at a section of skin, you'll often be able to, well, you won't see that. Merkel's discs, these are Merkel's discs labeled with a fluorescent antibody. So this green stain is labeling the axon, and then this orange structure is the actual Merkel's disc. A Pacinian corpuscle has been likened to an onion cut in half. So it's this layered structure here, very distinctive. And then Meissner's corpuscles. And I've described these as slow or fast. And what I mean by slow or fast is that they're slow or fast adapting. And this is really key. So a lot of this is sort of, like I'm just sort of up here talking, but the slow and fast adaptation is really key. And this is just how quickly they adapt and stop firing after a stimulus. So we have fast, and slow. And then they also are sensitive to different levels of vibration. So if you can think these are vibration detectors, those are some of their sensitivities. So this is an example of slow versus fast adapting receptors. Our stimulus is here. It's mechanical displacement, so that's a mechanical displacement, right? You always thought that was a touch, but really it's a mechanical displacement. And if an adapter is slowly adapting, what's going to happen is you have that touch, it's going to fire a burst of action potentials. Remember, this is our train of action potentials. It's going to be a little more intense at the outset. Then it's going to plateau off, and you'll have a fall off when the stimulus stops. So that's what a slow adapting receptor would look like. A fast adapting receptor, on the other hand, it's going to turn on, and then it'll immediately turn off, and then it'll fire again at the end of the stimulus. So a fast adapting receptor will cause action potentials to be fired at the onset and offset of the stimulus, whereas the slow adapting receptors will continue to fire throughout. And we'll see that that's important. This sort of constant firing is important, for example, to let your body know where your limbs are. So you have to signal how quickly your limb's moving, but also where its final location is. And that final location is encoded in this plateau information here. So that's what we mean by fast and slow adapting receptors. Our interoreceptors that we're going to talk about are the muscle spindle stretch receptors, so just they stretch, they open up, and the Golgi tendon stretch receptors. So these will be in your tendons or your joint capsules. Again, they're just really simple. They're getting pinched or they're getting stretched, and when they're pinched or stretched, they're going to have a changed conductance, generally an increase in flow of sodium. In all of these cases, you have some receptor potential, that's the change in conductance, and if that's large enough, it can trigger action potentials. I talk about a couple types of muscle spindle fibers. You'll see this as you go through, maybe in later when we talk about skeletal muscle. They just differ in their sensitivity. Your Golgi tendon organs, which I'm not showing here, are going to detect things like muscle tension and the rate at which tension changes. So I just want to mention these because we're going to have an example later on. For now, I don't need you to know anything else except there's muscle and Golgi tendon organs, so muscle spindle and Golgi tendon organs, and their trip channels, and they're what's going to initiate action potentials. So how would we classify these? Are they nociceptors? Nope. Proprioceptors, yeah, good. So they're telling your body where you are. So now that we've had that little bit of basic overview, I want to use these different receptors to illustrate how the receptor potential can give you a sense of intensity, location, quality or modality, and temporal components. And then we're going to finish up with pain. So the first thing we need to consider is intensity. And you'll see as we go through the special sense organs, we're going to come back to intensity. How can you detect the intensity of sound versus the frequency of sound? But intensity, what we want to consider is an experiment where we're stretching a muscle to 3 different lengths. So we have 3 different lengths of stretch, and that's going to activate the muscle stretch receptors. So what our question is, how do we encode that? How do we encode that different level of intensity? And I'll just mention that we're going to use tetrodotoxin to actually block the action potentials. Because if you trigger an action potential, it's going to be this huge distortion, and you're not going to be able to see the receptor potential. The receptor potential is pretty small. The action potential is quite large, and if you were measuring it, it would swamp it out. So we have 3 different levels of stimulus shown here. So small, medium, large. So this is our stimulus intensity. This dashed line represents threshold, and then this plot would represent the receptor potential. So if you had a small stimulus, you might not even be aware of it. You might not reach threshold here. If you have a medium-sized stimulus, you're going to peak up and then plateau, and then a similar plot with a larger stimulus. So in this case, we're blocking action potentials with tetrodotoxin. So all 3 cases, the duration of the stretch is the same. Now, if we were to measure out our action potentials, so what we would actually see, and these I should get rid of the threshold line here. If these were just action potentials, the intensity would be encoded in the number of action potentials during that period of time. So intensity will be encoded by the number of action potentials in a unit of time.
That it's different from the receptor potential in that the receptor potential is based on the actual intensity of the stimulus.
Yeah, so the receptor potential will be graded depending on the intensity of the stimulus. And the action potential, remember, is all or none. So your action potential is all or none. So you cannot have a big action potential versus a little action potential. So the way we get around this is the frequency of the action potentials. So if we have a big stimulus, that will cause a bigger receptor potential, and that will trigger more action potentials in the same unit of time.
Is it proportional?
It actually is amazingly proportional in some systems. I think that actually the last slide that I decided not to use this year talks about that. And it's proportional in different ways in different systems and plateaus out in different ways. So the last slide at the end that we won't talk about this year will show that. So it's proportional to a point. So what we haven't discussed yet is this little peak, but we're going to get to it in a little bit. Nicholas.
Can you put that faint line under the receptor potential isolated with tetrodotoxin? That's supposed to be the above.
Above.
Faint line right there. That's supposed to be what
this would just be a representation of threshold. And so ignore that here, these should be stretched out more. So that's intensity. So it's really simple. So intensity is encoded by the number of action potentials, and the number of action potentials is dependent on the size of the receptor potential, and the size of the receptor potential is dependent on the size of the stimulus. So that's how you can go from something that's graded to something that makes sense, that's really binary. So the action potentials are really it happens or it doesn't happen. Sure.
And the number of action potentials, is that just during the time that the stimulus is being, like you're being touched or something's being stimulated, can it have like action potentials after, and do you count those?
So this is a good question. So the question is, well, when the stimulus falls off, what happens? So in many cases, actually, at a baseline, you have a few action potentials. Every now and then you have an action potential. And then when you have a stimulus, you have a denser train of action potentials. You have more action potentials. And then actually when the stimulus falls off, you'll often have no action potentials. With other receptors, so for example with the fast receptors, the fast adapting receptors, you have the stimulus, you have action potentials, then you have no action potentials. And you have no action potentials until you have no stimulus, and then you have action potentials again. So there'll be a change both at the beginning and the end. And we'll see some specific examples of that with like the Merkel's discs and Pacinian and Meissner's corpuscles. So next up is coding of location. This is really simple. This is basically just where the receptors are. So they're mapped out somatically. So if we could think here, you have different receptors, and they're going to send information to different parts of your brain. It's where we get this funny-looking person here called a homunculus. So the homunculus is sort of the little person mapped onto, for example, your motor cortex, and different regions of the motor cortex or cerebellum or whatever else have different amounts of space depending on how much of it is devoted to a different part of the body. So for example, our hands get tons of representation in the motor cortex. Our lips get a lot of representation in the motor cortex. And if you were to map out proportionally the size of it, you get the homunculus. So that's where that comes from. And you have different homunculi for different systems. You'll have your motor cortex and your different sensory systems as well. We'll map out. Now, what this means is that different parts of your body, of course, are differentially sensitive. And this will be different for different systems, but generically you have something called two-point discrimination. So if we're talking about touch, we have two-point discrimination, and that's just the point at which you can tell that two stimuli are actually two stimuli. This is very small, maybe for our fingertips. It's very large for something like our back. Our back is like 5 centimeters, so 2 points, you wouldn't tell that that is 2 separate stimuli. You would detect it as a single stimuli. So if you think you have a bug on your back, right? When you have a bug on your back, you never like take one finger and are like, plop, right? You take your hand and you go whack, because you don't really know where it is. And that's because your two-point discrimination is really pretty bad on your back. And this is just this mapped out. This is millimeters. So you can see our fingertips, our lips are very sensitive, but other parts of our body are not they have a very small two-point discrimination, whereas other parts of our body, this would be spaced much further out. And this is just the receptors are more spaced out. They map to smaller areas, so more of them would map to a single area in the brain, and you actually just can't detect whether that's 1 or 2 points because they're all in the same field. So this would be encoding location. Next up is modality. Everyone okay with location? All right, so modality is just, it's almost just the adequacy of the stimulation. So your different receptors, you can think they're sort of fine-tuned to detect something. And that's what they're good at, but if you smack them, they're probably going to respond. So if you push your eye, you're going to stimulate your ganglion cells, and they're going to fire, and it's going to look like you're seeing light, but really you're just activating the cells that tell your brain that you're seeing light. It would be similar phenomena with reference pain. You might have pain in part of your viscera, and it's you know, the classic would be a cardiac event, and you feel pain in your Nicholas.
What causes like ghost pain?
Ghost. So like if you have your limb
Yeah.
amputated, you have all the upstream structure there in many cases, and so even though you don't have the receptors there, you have the other structures, so those can still be activated. It's really an interesting, a really interesting area of, I would say, research or phenomena, because it's almost like if you think you have the hand there, you won't have it. So if someone has the hand amputated and you sort of project out the hand, then your brain makes it go away. So what about our psychologists in the audience, Jacob or John? Do you go over this in more detail? Am I putting you on the spot?
Yeah, it's so like being a trauma challenger, did a lot of research on it. It's like you said, it's the upper level kind of feedback processes that some innovation happens, after the damage to the brain, where those cells start to die off and spread out a little bit. So, both momentums in the brain. Did you?
And you explained it quite well. Just local stimuli of it basically activates those nerves, and so you feel that the arm is still there. But when you do show like a mirror image of it or a projection of it, and you alleviate that pain by, let's say, just giving the projection of it a massage, then your body.
Yeah, so that's the part I think so amazing. And I mean, it's got to be some higher order level of processing that's doing that. So somehow your visual system's feeding in. So some examples of this, some examples of adequacy of stimulus. I've mentioned poking your eye. Another would be paradoxical cold. So heat above a certain level can feel cold just because you start to damage those receptors and activate them. So it's not necessary to have the ideal stimulus to get a sensation. It's really if you're activating that receptor in a number of different ways, then you can get some sort of stimulation. So how is this relevant to our case study? What was our patient encountering? Tingling. Yeah, so is someone actually, like little gremlins are tickling his feet or something like that? Probably not, yeah. So there's some sort of nerve damage that we'll discuss at the end that's actually causing that tingling. And so it's really diagnostic actually for what stage of damage has occurred. So next up is temporal components. This is a little bit more complicated, and again, it's going to involve our slow adapting and fast adapting receptors. The slow adapting receptors are going to be key though for encoding how fast something is occurring. And there's a classic experiment by Boyd and Roberts where what they're doing is they're moving a joint a certain number of degrees per second and then ending in the final location. So somehow your brain has to know the speed at which you're moving and your final location. So that's what this experiment sets out, and you get to see my wonderful PowerPoint art. This is a leg. This is a foot right here. It's awesome. And the experiment is eventually you want to have 90 degrees of stretch. But you're going to apply the stretch at 6, 10, or 17 degrees per second. And you have your degree of stretch, and this is what the receptor potential would look like. So again, you're blocking any action potentials with tetrodotoxin and recording this. So you actually wouldn't do that in a patient. So you have your duration of stretch here and what your receptor potential looks like. So initially you're going to have a little spike in conductivity. So your receptor potential, remember, is change in conductivity, and then it's going to plateau out. And if you're moving more rapidly, you'll have a bigger spike, but then you'll plateau out. So you're adapting to the stimulus, but we call it slow adapting because it's not like the fast adapting where you have a few action potentials and it stops. So you have your action potentials, and then you plateau out, and the intensity is going to be encoded in this initial burst of action potentials that are associated with this. So imagine, when you first stretch open these receptors, they're going to put through a few more ions than at sort of the level state. So when you initially activate them, they're going to send a little more stimulus than in this plateau, and the result is going to be that you're going to have a more intense burst of action potentials at the onset, and that's going to encode speed, and then you'll hit a plateau. And then when you reach this plateau, you're going to keep sending action potentials at some rate, and then when you put the limb back in its place, those action potentials would fall off. So this is how slow adapting receptors can give you both intensity and location. Sure.
If you're varying speed of stretch.
Oh, I knew someone was going to ask that. Yeah.
So are you going to see like multiple spikes like that initial spike?
It depends on whether you're speeding up or slowing down. So imagine you could be going quickly and then slow down. So remember, I mentioned that when you stop the action potential, you'll often go from, where you have a few action potentials now and then to just none. And so that could be used in the plateau if you were, say, slowing down, for example. And if you were speeding up, then you would hit a few more and you would have another spike. So it depends on the increment versus decrement.
Okay.
Good. All right, so this is just this again in another slide. I like to sometimes make the same slides, and that's why I like to make summaries too, so you're not like, what's the difference between these two? Here we have our stimulus, and we have our receptor potential, and then this would be what the action potentials look like. So each of these lines would represent an action potential, and that's how the chain of action potentials are depicted, and so you'll see those more often now in class. All right, so even though your stimulus is constant, you're going to still plateau out and be firing some new rate of action potentials. And for any given limb, that's how your cerebellum, that's how your brain knows what the location is, because it's sending a certain speed of action potentials. And then if you change that, you'll have a spike or a decrease in the number of action potentials there. So slow adapting receptors are going to encode the duration and intensity of the stimulus. And everyone remembers what our slow adapting receptors are, right? So everything except our friend, the Pacinian corpuscle. Pacinian corpuscle, Meissner's corpuscles, they're fast adapting. And the way fast adapting receptors work, you have a stimulus, you apply a stimulus, you get a short, dense train of action potentials, and then they are fast adapting, so they stop. You don't get that plateau of action potentials. You get another train of action potentials when you turn the stimulus off. What this is good at is encoding the frequency of stimulus. So how often is a stimulus happening? This sort of fast adapting receptor is able to encode that. And this is just what a Pacinian corpuscle, what its firing would look like without that capsule, so without that onion ring around it. It actually would just kind of fire tonically. So if we say it's firing tonically, we mean just sort of constantly, just like that plateau. So the actual structure of that receptor is what makes it fire initially and then at the end, at the offset of the stimulus. So slow and fast adaptation, a couple examples of each. Everyone good? Should we take a break? Do pain? Do I have my pain intro? I do have my pain intro. All right. So Meissner's corpuscles can tell you about a moving stimulus. So if you have a little bug crawling on you, it can tell you the direction of movement just because they're mapped out across the body. And then the bug bites you and you feel pain, and we'll talk about that in 10 minutes.